Hello everyone, this is your teacher Mrs. Namira Zahid and we are here together to learn something related to inheritance. We were talking about the structure of DNA and how DNA is present in the nucleus of the cell. So you can see this is a cell and this is the nucleus of the cell. When we are taking up single chromosome, this chromosome is made up of DNA and here you can see the nucleotides which are making up hydrogen bondings with each other. This is the segment of the chromosome, which is considered as, the segment of the DNA is considered as gene, and the DNA supercoiled structure, which is actually the hereditary character, hereditary material. It transfers the hereditary characters from parents to offsprings. Okay, so this is how DNA looks like, and we have already discussed about the structure of the DNA. In this way, we can say that there are so many different parts of the DNA. The DNA is consisting of the whole molecule. It is consisting of a long strand. It is supercoiled. It is double-stranded. It contains 4 nitrogenous bases, that is adenine, cytosine, guanine, and thymine. And the DNA strands are complementary to each other. In this picture, you can see that the chromosomes are containing supercoiled structures of DNA, and DNA is actually the structure which is supercoiled, and these coils are kept together by histone proteins. Okay. This is how ladder-like arrangement of DNA is explained through Watson and Crick model. And this ladder-like arrangement is twisted and supercoiled to make it a helical structure. This is again a DNA model project which shows the basic uprights. These are the uprights you can see, and these are the rungs. This part is called the upright, and these are called the rungs. Okay. This is how DNA looks like in molecular arrangement. Now, in this lesson, we are going to talk about the genes. What are genes? Genes are the parts or the fragments of DNA containing useful coded biological information. Okay. So gene is a small segment of DNA which has genetic information in the form of a code, and it can synthesize a protein. It is the basic unit of biological information. So we can define it as the basic unit of biological information. Each gene is located at a specific position on the chromosome, which is called locus. Okay, the specific position of a gene on a chromosome is called a locus. Okay, that is the position of that specific gene on a specific chromosome. Each gene has a specific function. It determines the height of the plant. It can determine the color of the petal of the plant, and any gene, each gene has a specific function. We can consider any gene which has a specific function. So each gene determines a specific character. Okay. So it is responsible for a single character or a specific function. Okay. Now, the coded information may change due to any reason which may be called genetic variations. So what are mutations and what are genetic variations? We can see the genetic variations, which are also called mutations. What are mutations and how are they caused? So sometimes it happens that due to some kind of radiations, which can affect radiations like X-rays, UV rays, okay, or some other mutagens, like these can be the chemical mutagens, which have the ability to change the sequence of the genes. Okay, when the sequence of the genes is altered, it is called mutation or genetic mutation. Okay, so the coded information may change due to any reason, which is also called genetic variation. It is caused by mutation. This genetic variation among offsprings led to tremendous variety in their traits, and this variation gives rise to two or more than two alternative forms of a gene. These alternative forms of genes are called alleles. These alleles are the genes which are regulating the same character. So what are alleles? Alleles are the genes or set of genes which are regulating or controlling a specific character. Okay. What is a specific character? Like, for example, eye color, there are so many genes, one gene for green eye color, one gene for blue eye color, one gene for brown eye color. So the set of genes which are controlling the eye color are called alleles of each other. So some genes may have as many as 300 alleles, and a diploid cell, thus any two of these multiple alleles with a haploid have one allele, right? So genes are basically considered as the unit of inheritance, which are copied and inherited to the next generation. Why genes are important, why the knowledge of genes is important or useful? Just because when we talk about the complementary arrangement of nucleotides, it is of great importance in DNA molecule, because if the sequence of bases in one strand is known, the sequence of bases in the other strand can automatically be known, right? So this is called base pairing. And the property of two strands of double helix makes DNA a unique molecular structure or a unique molecule best suited to store, copy, and transfer genetic information from generation to generation. Like you can see that this is a DNA molecule, and this DNA molecule is double-stranded, and the two strands are having complementary bases. As we know that adenine will always pair with thymine, and thymine will always pair with adenine. Similarly, cytosine will always pair with guanine, and guanine will always pair with cytosine. It means that DNA is actually having a complementary arrangement. Okay, so this is the complementary arrangement of the two strands. In this way, we are able to find that the sequence of bases in one strand will determine the sequence of bases in the other strand, and the two strands are complementary to each other. Now we know that the DNA is able to make its exact copy itself in the nucleus before cell division. As we know that cell division has many different phases. Cell division has one phase which is called division mitotic phase, which is the division time. We can see that there is an interphase which consisting of S phase, which is called the synthesis phase. Okay, and other G0, G1, G2 phases, which are the gap phases. So this phase, synthesis phase, is really an important phase during which the DNA replicates itself inside the nucleus of a cell. So DNA is able to make its exact copy itself, and this occurs before cell division. The process of duplication of DNA is called replication of DNA. Gametes are haploid cells carrying one copy of the replicated DNA, and this copy carries gene from parents to offspring through the gametes. Okay, so DNA replicates before cell division. This DNA replication is actually important, and it is really important. It is really necessary for the formation of gametes. The Watson and Crick model of DNA ideally proposes the mechanism of self-replication of DNA. And it is when DNA replicates itself, it is called self-replication of DNA. And the DNA actually replicates itself when the hydrogen bonds which are holding the double helix of DNA are broken up from one end to the other end, like unzipping. What happens? DNA unzips at one end, okay, and the complementary strands are forming against each strand. When the helix gradually unzips along its length, each new nucleotide of the complementary nucleotide would be added to separate chains sequentially, and it forms the replication of DNA. Like, for example, if it is like this and this, it is opening up, unzipping, and the new strand is going to form here with this strand, okay, and with this strand too. So these complementary strands will be forming. In this way, the DNA will be replicated. So in the next lesson, inshallah, I'm going to discuss about the process of replication of DNA in detail. And we'll also discuss about the central dogma of protein synthesis by genes. Thank you.